So at Diagonal Bio, we are creating the diagnostic platform of tomorrow. Our first product is called PanViral. It has the potential to be the fastest, high-precision diagnostic platform for testing of any type of pathogen. It can be an essential tool for the healthcare community. It can help ensure that we are better prepared. It can help to ensure that we can prevent and also diagnose. So PanViral detects genetic material in real time with very high accuracy and sensitivity using a simple one-step assay. So what we are trying to do is we are trying to take the gold standard of diagnostics and bring it to point of care. Essentially avoiding to go to these central laboratories, but being able to do it directly with the patients. The world needs high-quality diagnostics. I think that's pretty evident on top of this COVID-19 crisis. We need diagnostics which are accurate, easy to use, fast, available at point of care, and cost efficient. And that's exactly what we can offer with PanViral. So what is the problem today? Today, most of the diagnostic testing is done at central laboratories. That means long response times, risk of spread of infections, and delay in treatment. With PanViral, we can ensure point-of-care diagnostics without compromising accuracy and sensitivity. This means quick identification of the infectious disease at point of care. It means optimized treatment or treatment very early on, and quick isolation of the patients. Every single time you have a new outbreak, you're always one step behind in that we do not have rapid diagnostics to detect a new organism. With PanViral, that can change. Diagnostic tests are often the least expensive component of the healthcare pathway, yet they influence more than 70% of the healthcare decisions. Again, with PanViral, we can take the right decisions very early on in the process. Frequent and real-time tests are critical to reduce infections. So this is modified from a study that was done from Howard University and I think it was Ohio from 2021 based on COVID. So if you see the gray bar in the bottom, that if you have no testing at all, you have 100% spread of the disease into the group. What you can also see here is that more frequent you test, the more you can limit the spread of infections. Where it gets really interesting is if you look here. So even if you test every 3 days, if it takes more than 2 hours until you get the results, you can have up to 45% spread into the group. If you, on the other hand, can get the results immediately, then you can limit the spread of infections to almost zero. And that's exactly what we can offer with PanViral. This was a small exploratory study that we released in December last year on detection of COVID below 10 minutes. I don't want to go into too many technical details here, but what you can see here in the lower part of the graph is the negative controls. And then you see there's a threshold, the orange bar, and then you see the positive samples where we see an exponential growth. That's what we measure here. So in this study, we detected COVID samples on an average below 10 minutes. This is a recent study that we did this year, where we showed detection of 2 pathogens in parallel. In this case, we chose COVID again as a model organism and also influenza A. And as you can see in the data here, we detected this with a detection time very close to 9 minutes. These 2 organisms are just as an example. PanViral can detect any type of genetic material. This could also have been Ebola, it could have been sexually transmitted diseases or anything else. But we can do it in parallel as well. And I'll get back to that, why this is really unique. PanViral is a unique system. There's no other system out there but combine all of these three features that I will show here. Speed, so as I just showed you, we can detect below 10 minutes. We have an accuracy that resembles that of the gold standard for diagnostic testing today, which is the PCR. And we can test multiple samples at the same time. Again, whether this is multiple patients or different types of diseases within these 10 to 15 minutes. This is creating commercial opportunities for PanViral at a global scale. A little bit about the technology behind PanViral. We use a combination of Louvre-mediated Israel amplification, also called LAMP, and we couple that to electrochemical sensing. So the whole idea here is that we know a specific target within the pathogen genetic material, and we then amplify that exponentially. By coupling that to electrochemical sensing, we can detect that very early on and very precisely. This is a first industrial defined design file of PanViral. So what you can imagine here, this is a small box, basically half the size of a laptop. Of course, you have more depth. There's a very easy to use interface touchscreen in front. So basically everybody can use it. And what you can see here on this model here is that we have a panel inside. In this case, we have 8 channels, which means that this prototype will be able to test 8 different samples simultaneously, whether that is 8 patients or 8 different diseases at the same time. So what are the benefits? There's hardly any sample preparation needed for running this assay. It's really robust. We can detect any type of genetic material, whether it's DNA or RNA. It's extremely fast, as I just showed you. We can run everything with one single temperature, and I'll get back to that, also why this is important later on. It's very easy to use and customize. So basically, we need to change a little bit in direction mix to change it to detect another organism. Technically, that could be done in a couple of days or even weeks. It's cost efficient. We can cut away a lot of these sample handling steps. And also the hardware by itself is very simple by design, cutting a lot of costs. It's ideal for point of care. Again, it's an easy, small machine. It can be standing anywhere. Anybody can use it, basically. And as I said before, it's a universal system. Whether we're using PanViral for testing sexually transmitted diseases, Ebola, COVID, there's basically no limitations here. And then again, we can test multiple samples at the same time. You can imagine that when we get to the fall, there's a new mutation of COVID. We will be able to screen for different types of COVID mutation, influenza A and B, and respiratory bacteria, within these 10 minutes, having the same accuracy as with PCR. And the technology, of course, is protected. So we have a patent pending technology with a priority date in March 2020. This is very basic to describe how you normally do, you can say, the principles behind gold standard nucleic acids amplification as it is today. So first of all, you need to know your target. Then you remove everything that you don't need. Very often it will inhibit the reaction if you don't do that. Then you need to add resources. You find your target, and then you amplify it, and then you analyze it afterwards. So if you compare to how that works with, you can say, the PCR-based technologies today, again, you need to know the pathogen that you're looking for, the genetic code. Then you need to remove the excess material that could inhibit the reaction, so you do a purification step. You need to add enzymes. Then you add fluorescent primers. And then you need to go to thermocycling. So that means you need to go up and down in specific temperature steps in order to amplify the target. And then you have optics inside the system. So you have LEDs and other types of advanced electronics to actually detect the target. If you do the same comparison with PanViral, then we still need to know the genetic code. We can skip the purification steps. It's a very robust assay, so you will be able to add a sample directly into the system. We still need to add enzymes. We also need to add primers. We We don't need to have this fluorescent primers. And then we need to add heat as well, but we only need one simple temperature. And then we need electronics to detect. So where are the benefits here? If you look into this, where we can avoid the purification step, that means a lot of automation, as it is today in these central laboratories, we can skip that step. That means we save time and money. For the primers, we don't need this fluorescent tag. That means that we save money. And we can also say it's better for the environment. Then for the thermal cycling, we don't need that. We just need a simple temperature. That means more simple electronics again. Again, we're saving time and money. And for the detection, the way that we detect, we have the electronics dip directly into the solution. So we avoid having these expensive optical systems. Again, making it fast, more reliable, and more stable. So if you try to list them here, we can do detection within 10 to 15 minutes. We can basically avoid sample preparation. We can reduce the hands-on resources. That means time and effort. Because it's a more simple system, we can reduce failure by design. We can reduce the amount of software or hardware that goes into the system. And that also means we can reduce the complexity of manufacturing. We can eliminate optical hardware. And we can eliminate these fluorescent or toxic components that you would normally need. So if you benchmark PanViral up against the gold standard of diagnostic testing today, which are these PCR-based technologies, then most of these systems, just running it on the PCR machine, will take a couple of hours because you need to go through these different temperature cycling steps. The whole process with our system, PanViral, can be done in 15 minutes. And that's from taking the excitement to actually getting the results out of the machine. If you look into the mass testing capabilities, as I just showed with the prototype I showed you here, we had 8 channels. We have also had prototypes that had 96 channels. So again, with PanViral, we enable mass testing potential. And the last one here I want to show is accuracy versus, you can say, costs. And because we can cut a lot of these sample handling steps away, we have more simple hardware. We can offer the same accuracy at much reduced cost. Looking into the infectious disease testing market, it's currently valued at $34 billion US. And that's not taking into account this COVID situation we just went through. The market is dominated by these quick tests. As you probably know, they are not that reliable, but they're very easy to use. Then we have the PCR-based technologies. And as the third one, we have these CRISPR-based technologies, which is a very capable system, but also very complicated and expensive. So what problems do we solve with PanViral? First of all, today it's a trade-off. Either you go for the accuracy, then you go for these PCR-based technologies. They're expensive, they're time-consuming. Or you go for the easy solution, which are these quick tests. They're very easy to use, they're cheap, but they're not very reliable. The second problem is that most of these tests today, they're very diagnose-specific. So you only look for one component at a time. Our solution, PanViral, have the capability to be the fastest, accurate, cost-efficient system on the market for testing multiple different pathogens or multiple patients with extreme ease of use. There's simply no other system on the market today that can do what we can do with PanViral. If you look into the market niches, then because PanViral is a universal system, there are so many opportunities out there. Where we see a good fit could be for tailored regional specific panels. And as I said before, when you come to the fall, there will be a new COVID mutation. We will be able to make a panel where we screen for different COVID mutations, influenza A and B, and respiratory bacteria within these 10 to 15 minutes. Could also be that you want to travel somewhere exotic, and before you enter into your country again, you get tested with this at a tropical panel. It could also be used for tests for Ebola. I mean, there's basically no limitation here for Panviral. You can also use it as a state-of-the-art outbreak system. We have very high sensitivity on the system, so even before you show symptoms, we will be able to detect that with Panviral. And the latest one I will talk about here today is about disease and treatment monitoring directly at the hospitals. Because of these real-time capabilities for Panviral, we will be able to validate treatment directly at the hospitals. Very briefly about the commercial plan. So overall, you can say this is based on the go-to-market strategy here is based on this razor blade model, or the printer model that we just heard about before, where you can say the main driver for the revenue will be the kits. So these different panels of specific diseases you can scan for, these are consumable and one-time use only. So that will be the main driver of the revenue. Very briefly about the milestones and financials. The company was founded in Q1 2020. That's also where we have the priority for the patent filings. We did a small study on clinical samples from COVID patients to show that the technology is working. And with that, we brought in 9.5 million from private investors. Since then, we tested the system on different bacteria, viruses, and fungi to show the robustness of the assay. We then listed at the Nasdaq First North in July last year, and we brought in around 40.5 million. We have done proof of concept of our new prototypes. We have implemented our quality management system and are looking into the regulatory process. And the plan is to start the commercial activities this year. We don't plan to go all the way by ourselves. We want to partner up with somebody who already have the distribution channels and a way where we can get out there in a, you can say, fast and efficient way. Very briefly about the team. I didn't get a chance to introduce myself, so I can do that now. So my name is Jack Egelund Madsen. I'm the VD for Diagonal Bio. I have a technical background with a PhD in biotechnology and have been working as an associate professor at Copenhagen University, but in gene discovery. But I also have a commercial education from Copenhagen Business School within business administration. And before joining Diagonal, I was working in different commercial and managerial roles in pharma, CRO, etc. We have Per Hender as the Chief Financial Officer, Andreas Nyberg as Chief Scientific Officer and also one of the co-founders. Then we have Jay Shen as a Product Development Leader, Roberto Monita and Roman as a Research Scientist. We have Stefan Magnussen as our QA Manager, and Caroline as our Grant and Office Manager. We have Emily as our Lab Technician, and then the latest addition to the team here is Joachim as our RE Manager. Then we have an external HR and also an external consultant helping us on our quality management system. We also have a very strong board. It's, the chair is Ivan Nilsegård, but we also have Kerstin Jacobsen, former CEO of Medical Village, Edward Hall, CEO of BioExtracts, and Kusukar Punyani, co-founder and also main, could say, inventor behind the technology in the board. So that I would end up by saying our vision is to limit the spread of infectious disease worldwide by creating an easy-to-use, accurate, cost-efficient, and quick universal diagnostic platform for testing of multiple infectious diseases on multiple patients in parallel. And with that, I would like to open up for questions.
Thank you, Jack. Okay, just to a dumb question.
Yeah.
This Panviral, how far along is it? Do you have it? Do you have the machine performing at this PCR accuracy, or do you aim to have it? Is that a goal?
We have iterations of the prototypes that we've been working on. So we have had multiple prototypes, physical machines that we've been working on, and we're of course working very hard now on to get something that's not just a prototype, but something that's a commercial version.
Okay, because this, is there a risk that you only come to, I use COVID, the antigen accuracy, which appears to be low? Are you certain you will reach PCR accuracy?
That's why we are doing all the tests that we have done so far instead of just going straight to putting this into you can say, a commercial system. So we are taking all of these precautions and finding the most accurate, you can say, technology before we start manufacturing.
Okay. So you have, can we use the back button and go to the slide where you have the bars with the 3 days testing interval?
3 days.
Just to check some basic understanding. It's in the beginning. There.
Not this one.
There. So, okay, yes. So here we see that because the accurate testing is in real time, we only need to apply this test every 3 days. If we did that in the Olympic Village recently, we would have caught almost all positive results and been able to isolate them. Is that correct?
That's correct.
Okay.
Yes. But of course, this is a study. I mean, this is not real-time setting. This is a small study they did.
Yeah,
of course. Yeah.
And that was not available now. Then they had fast but inaccurate antigen testing, or accurate but slow PCR testing.
Yes.
Okay.
A couple of questions. Can you improve accuracy from 96.5 to say 99%?
We are working on that, and I can also say these results, as it was also stated here, was from last year on a different sensor prototype. And we have developed a new one that we're working on right now, and we're expecting to come out with data later on with updated accuracy.
Can you examine blood samples?
The assay is pretty robust and should be able to take all different types of matrices. And this is also something we will go into, so we can give you, you can say, a study where we can actually show different types of matrices. So that's also ongoing.
And can you differentiate between different bacteria?
It's not the way, if we can, it's like with a PCR. So if you know the genetic code, you can find specific regions that differs between the different pathogens, then we should be able to differentiate them as well. So basically the same principle as you will see with a PCR.
Because if I had a blood sample, I would be interested to know more or less what sort of bacteria it is, because then I can guess what antibiotic is suitable. So, I mean, treating within a few minutes is essential.
And of course, this, as I said, we can detect basically anything with genetic material. And if that was to test for antibody resistance genes, for example, we can definitely do that with the system as well.
Yeah. The accuracy is to find when it's you have an infection, but do we have positive for non-infected also?
It depends.
Do you say that it is infected, but it's not infected? Because that's
you're talking about what we call false positive, I think.
Yeah.
Oh, yeah. You can, of course, if you have contamination in the lab, then you would, of course, also amplify, I mean, the contamination.
But have you found that in the study?
In which study?
Don't you have a study to come up with the accuracy numbers?
That was the first study that I showed you. Yeah. So there was some false positive, but that's more related to contamination in the lab. So of course the method is very sensitive. So if you contaminate and you don't keep everything clean, then you would have a false positive.
Just a quick 2 questions. Is there any formal requirements for the operative person who is doing the operation and doing the handling the machine? That's one question. And then when do you think you're ready for a pilot in Germany?
In Germany?
Yeah.
That was very specific. The last one I can start with that I cannot disclose right now. What I can say is that, of course, the machine is going to be manufactured under ISO 13485. And of course, we are looking into the IVDR, so we can get a CE marking under that. But there's, of course, other routes to market than that, and that much I can also say.
Okay, but is there any requirement for the person who's handling the machine?
These exploratory studies that I showed you here today, there was no formal requirement. So of course you need, you can say, general lab training. So these people are, they have a scientific career and are used to working in a lab. So it's not just random people who's doing this.
Yeah. We can't have too much without the microphone. Yes. Sorry.
Yeah, I have a question on the sensors. Are they in direct contact with the sample, or are they disposable?
It's disposable, so they will be part of this consumable part. There will be a sensor directly into the chamber where you have the reaction going. So that means we measure directly with the sensor in the sample.
Okay, thank you.
And it is a disposable. You use it one time and then you need to throw it out.
Quick one. No.
Just alluding to the question in the back, if I am a GP, allmänläkare, could I take a sample from a sore throat, get an answer within 15 minutes, whether it's streptococcus infection or a virus infection, or do I have to have some sort of a lab?
The whole benefit of this is that you don't need to ship it to a central laboratory. That's the whole benefit. So you still get the same accuracy. And again, if there's a genetic marker for that, what you're talking about here, then definitely yes, not a problem.
But you have to, your machine cannot tell us what type of problem, infection the person has. It can tell us, does this patient have infection XY2?
Yes. So for example, we have this with 8 channels, and you can say we will screen for 8 different types of diseases in those channels, and you will get a result whether that's positive or negative basically for these 8 diseases that you then chose. It's not like you can just ask any question. You will need to know what you're looking for first to find it.
You have a very specific target, that is to reach break-even, and I think you mentioned what looks like a low figure, 32 million in 2023 for sales. Why did your company come up with that number? It seems like it would be another 0, but maybe it takes 4 or 5 years. But you set a pretty low number and you set a pretty short time frame.
We are very ambitious, and as you also can see here, this really has global potential when we go out there. But again, you cannot expect to, from one day to the other, to actually transform the whole industry. That will take some time. So that's why we have been, you can say, not setting the numbers too high.
Yeah. So if you reach 32 million for pan-viral sales 2023, when will you reach 320 million?
We have worked with different projections, of course, but I don't want to say anything here, and then you keep me on that. So I think that let's wait before I come with a statement like that.
That's pretty smart. Okay, thank you. Diagonal Bio.
Thank you.
Jack, thank you very much.